Hello, I'm Nick Ruszala from Bio 200. This is my lecture on protein structure. Okay, so proteins, to start off, are one or more linear chains of amino acids. Now before we start talking about the actual structure of proteins, you need to figure out what an amino acid is. An amino acid is a molecule that is made up of 4 different groups. It's made up of a carboxyl group, which looks like this, a central carbon, which is always connected to a hydrogen atom, a amino group, which is a nitrogen atom connected to 2 hydrogens, and an R group. Now, the R group can vary on many amino acids, and the R group is actually what determines what an amino acid is. There's 21 different types of amino acids, but I'm not going to go into that, so I'm just going to write R. Okay? So that's an amino acid. Carboxyl group, central carbon, and an amino group. When you actually get into proteins, it's a bunch of amino acids combined into things called polypeptides. Now these are basically just long chains called polymers of these amino acids. And these are formed through peptide bonds. And for each of these chains of polypeptides, there is, on the end, an N-terminus group with a positive nitrogen atom and 3 hydrogens. And this connects to a chain of amino acids. I'll just write A for amino acids. And this is just a chain of amino acids that are joined together by peptide bonds. And on the other side of these chains is a C-terminus group, which is a carbon double bonded to an oxygen, and then with a negatively charged oxygen hanging off the side. Now this is basically a drawing of the first type of structure of a protein, the first level. There's 4 levels. This structure is called primary structure. Basically what this is the amino acid sequence of a single polypeptide chain. Now this is controlled by the amino acids that are in the middle. Now each of these proteins has a very, very unique primary structure. It's what makes the protein separate from other proteins. And so basically it defines what a protein is. Now this structure is held together by peptide bonds, which I said before are covalent bonds. These are very, very strong, and overall primary structure is the strongest of all 4 levels of the structure. It's very, very hard to break these down. You're going to need an enzyme or a very, very strong acid to break these down. Now, moving on to secondary structure. Secondary structure is basically the localized area of repeating structures in proteins. So basically, there's 2 different types. There's an alpha helix and a B-pleated sheet. So basically how it works is that another one of these peptide chains, I will draw, and there's the hydrogen, hydrogen, and hydrogen, positive, and there's another C-terminus group and amino acid. Basically what happens is these 2 polypeptide chains are joined together by hydrogen bonds, which I'm going to represent with this orange line. These are hydrogen bonds. And these hydrogen bonds alone are usually very weak, but since there are so many hydrogen bonds in these structures, it's relatively pretty strong, but not as strong as a primary structure. And where these are held on the amino acids is basically, it's going to be on the carboxyl group of the amino acids. It's basically going to be where between an NH and a carbon double bonded to an oxygen is where these hydrogen bonds are going to end up. It looks like this, and it's usually signified by the Greek letter alpha. It kind of looks like that, but excuse my handwriting. Alpha helix. And there's also a beta pleated structure, which looks somewhat like this, but yet again, this is just a rough drawing they don't actually look exactly like this. And these are basically pleated sheets of these peptide chains is how it looks. And yet again, you can see that hydrogen bonds are going to form all about these structures through here, through here, through here, through here. And so there's a bunch of hydrogen bonds, which makes it relatively pretty strong. Okay, next we're going to move on to tertiary structure. Now this is the final 3D structure of a single polypeptide chain. And in this, there's a bunch of different bonds, but they're all relatively weak bonds overall. You're going to have the hydrogen bonds. They can form anywhere between like the side chains. You can have ionic bonds if you have charged amino acids with charged R groups. That means there's going to be specifically charged atoms on the R groups, and then they're going to form ionic bonds because, as most of you probably already know, ionic bonds are between two differently charged atoms come together. And these are relatively weak because it's going to be in an aqueous environment, so it's going to be pretty weak. And then you'll also find some hydrophobic interactions and van der Waals interactions, which are very, very weak. They're the weakest kind of bonds you can have. But there can be covalent bonds if you have two sulfate atoms that happen to join together between R groups. Now this can be very strong because it's a covalent bond, which is the strongest bond, but it all really depends on the R group of the amino acid. So finally, we're going to move on to the last structure, which is the quaternary structure. Now this is the final 3D shape of two or more polypeptides. So it's going to be two or more of those tertiary structures coming together in one to join as one big overall structure. Now they're going to have the same exact bonds as tertiary structures. They're going to have they're going to be relatively weak. They're going to have hydrogen bonds between the side chains, ionic bonds between charged atoms, and hydrophobic interactions. And if they're lucky, covalent bonds if there's two sulfate atoms. Yet again, relatively weak unless they're covalent bonds, then they're pretty strong. Now basically, what the quaternary structure is it's when proteins are actually functional. I'm going to give you an example. I'm going to do somewhat of a drawing of hemoglobin, which is a tetramer, which means it has 4 different of these polypeptides. So you're going to have 4 different of those long, very diverse tertiary structures coming together in one. So it's going to be all squiggled up and random. This will be one of them, and then you'll have another one joining. And you'll end up. This one happens to have 4, so there's going to be 4 of these coming together. And proteins usually do not end up coming in the perfectly folded states, the perfect structures. It usually takes some chaperone proteins to help fold them and form. But overall, the final quaternary structure is going to look something like this. Well, not really like this. It's going to look like this on a very, very small level. It's just a bunch of different polypeptide chains joining together to form one big mass, which is going to be the final shape and when proteins can actually do their functions. All right, so yet again, the primary structure, which is just the definition of a protein, it has very strong covalent bonds between, it's very strong. Secondary structure, you're going to have those hydrogen bonds, but there's a good amount of hydrogen bonds, so it's relatively strong, but not as strong as primary structure. Tertiary structures, which is the long joining of the polypeptide chain, the 3D structure, and that's going to have all different sorts of bonds. They're relatively weak unless you have covalence. And quaternary structure, which is tertiary structures coming together to form one giant mass where protein can have its function. Same exact bonds as tertiary structures, not too strong, but they get the job done. Thank you guys for listening. Hope you all have a good day.